Okay, very good morning, my dear friends. So this is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. So in this regard, we would like to invite our respected resource person, Dr.A.Shailaja Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Siddipet, Autonomous. Madam, please welcome. I have given a share option. Please, you may proceed.
Okay, sir. Thank you. Thank you very much. So today our topic is male gametophyte, development of male gametophyte. So up to anther structure in yesterday's class completed. So today we are going to discuss the development of male gametophyte. It's very important. Okay. Generally, that in angiosperms, the microspores represent the beginning of the male gametophytic generation. After release, the microspores from the tetrads are referred to as pollen grains. The microspore, that pollen grain, is the mother cell of the male gametophyte during the gametogenesis. So, in that, the nucleus of pollen grain divides into a small lenticular generative cell and a large vegetative cell. See here. So, this is a pollen grain structure: exine, intine, and nucleus, and germopore. So after that, this dividing nucleus, that is vegetative cell and generative cell. Okay. So this pollen grain nucleus of pollen grain divides into a small lenticular generative cell and a large vegetative cell. Okay. Then the generative cell again divides and form two male gametes. This division may take place either inside the pollen grain or in the pollen tube. So that is the, see here, and structure of pollen grain. The mature pollen grain is surrounded by a well-defined wall. So in the pollen grain structure, we will observe that mature pollen grain is surrounded by a well-defined wall, which has differentiated into two layers. The outer layer is called exine and the inner layer is called intine. Okay, this is the pollen grain structure. I am explaining about the pollen grain structure. The exine is the first appears as a thin layer, but later it becomes considerably thick. Okay, that is the exine. Outer layer is called exine. First, that is appears as a thin layer, but later it becomes the considerably thick. It can be differentiated into two layers: outer exine and inner layer exine. Again, that exine is differentiated into two layers. Outer layer is called as exine, inner layer is called an exine. Don't confuse Nana. So what is the pollen grain structure? We observed the two layers. That is the outer layer is called exine and the inner layer is called intine. Okay. So in the exine, again it can be differentiated into two layers. That is the outer is the exine, inner layer is the nexine.
Madam, is there any issue? Madam, your voice is not audible. Hello?
pollen grain structure. In the pollen grain structure, we are observed that exine and intine. Okay. అక్కడ వరకు విన్నారా?
Yes.
Okay. So that first layer is called outer layer is called exine and the inner layer is called intine. Okay. So then next that the exine it is a thin layer. Actually, it appears as a thin layer, but later it becomes considerably thick. It can be differentiated into two layers. Outer is exine and the inner layer is nexine. Again, the exine is divided into two layers. That is, outer is called sexine. Yes, sexine. Inner is called nexine. Okay. So this sexine is further differentiated into three layers. That is, basal one is called foot layer. A middle one is called bacula, and upper roof like it is called tectum. Okay, tectum, bacula, and foot layer. So these are the three layers we are observed in the sexine layer. Okay. Then the exine is provided with the small apertures or pores. These are thin areas where the exine is completely absent or represented by end exine only. So that the aperture is associated with the germination of pollen grain. So through the aperture, the intine comes out and forms the pollen tube. So when that intine is the inner layer, it comes out and forms the pollen tube. See here, I will show you in some diagrams. Okay, so it this is the pollen tube. Intine comes out and forms the pollen tube. The exine can be differentiated into two layers. That is called an exine 1 and an exine 2. So then next we are coming to the development of male gametophyte. The first mitotic division in a pollen grain results two unequal cells. That is two unequal cells. That is the smaller one is called a generative cell and the larger one is called a vegetative cell. So that the larger one is called vegetative cell, which eventually forms the pollen tube. The smaller one is the generative cell, which forms two male gametes by another mitotic division. See here, remember this, it's very, very important. Vegetative cell forms the pollen tube and the generative cell is formed as the two male gametes. Okay, it's very, very important. From the generative cell, the two male gametes are formed. Vegetative cell form as the pollen tube. Remember these two important things. Initially, the generative cell is attached to the intine of the pollen wall, but later it gets detached from the pollen wall and lies free in the cytoplasm of the vegetative cell. So then we are coming to the vegetative cell. The nucleus, the vegetative cell. The nucleus of the vegetative cell is spherical or irregular in outline with thin chromatin material. It has one or two nucleoli. The cytoplasm of the vegetative cell contains ribosomes and endoplasmic reticulum. All organelles are formed in this. Plastids, mitochondria, fat and starch grains. RNA is present in lesser quantity. Okay, the vegetative cell is normally does not divide, but although it is capable of DNA synthesis, that is the vegetative cell. Okay, larger vegetative cell. And coming to the generative cell, the generative cell is surrounded by a wall. There is no cytoplasmic connection between the vegetative cell and generative cell. The cytoplasm is relatively dense and contains usually cell organelles. So after that formation of male gametes, the male gametes are formed by the mitotic division of the generative cell. The division usually occurs before the pollen is shed from the anther. See here. So we are observing in this diagram that nucleus is going to mitotic division and form the two male gametes. Example is the beta and hardium. And in the former condition, the pollen are shed at the three-celled stage, vegetative cell and two generative cells. And in the latter, that is the two-celled stage, one vegetative cell and one gamete. So that some examples are there for that. In those plants where pollen grains are shed at the two-celled stage, the generative cell may divide after the pollen have reached the stigma. Okay. So for this example is Portilica or in the pollen tube. So in some plants we are observed in that male gamete in the pollen tube. Some are have reached to the stigma. So that some are reached to the stigma. Examples are Portilica, and some are in the pollen tube. Example is Euphorbia, Lilium also. Okay. So the two male cells of the gametophyte do have physical contact with each other. Such contact is important and helps in the coordinated development of both the cells. It is also important from the point of view of double fertilization. Okay, two male gametes are formed this like. This is the development of male gametophyte. Okay, all are you understood? అర్థమైందనా pollen grain structure and development of male gametophyte. From the generative cell, two male gametes are formed. అర్థమైందా? ఓకేనా? Please anyone respond. Shall I continue as megasporangium? If you have interested, I am going to next topic, that is megasporangium. Please.